This is part 2 of our chapter 20 recorded lectures. And let's see. Now we're going to be talking about prokaryotes, a little bit of a review on prokaryotes. The organisms which consist of prokaryotic cells are from the domain bacteria and the domain archaea. So prokaryotes are going to be unicellular, and they are going to lack a membrane-bound nucleus, although they still have DNA, which is found in the nucleoid region. Prokaryotes always have a cell wall. Some can move and some can't. They never have membranous organelles, so they're not going to have mitochondria. They're not going to have endoplasmic reticulum. They're not going to have Golgi apparatus or vesicles or vacuoles. Anything with a membrane, they're not going to have. Now, they will have one single chromosome, one single circular chromosome. But sometimes in addition to that, they'll have an accessory ring of DNA, a smaller ring of DNA called a plasmid. And we might talk about that. So bacteria and archaea are prokaryotes. That means they're unicellular and their cells do not have a nucleus. And what we're going to look at are the experiments that were conducted by Louis Pasteur. Louis Pasteur, and let's see if I can blow this up, experimented to see if he could use a different shaped flask to prevent the growth of bacteria. So in his first experiment, you can see he boiled the broth inside of a flask, and it had a stopper on it. So this was nutrient broth that contained bacteria. He boiled it to sterilize it. He opened the flask briefly outside of a building and let some time pass. And then 89% of his flask, like say if he did this with 100 flasks, 89 of them showed bacterial growth. So in other words, bacteria got into the flask from the air. So he showed that first in the first experiment. And the way that he detected growth is that the broth became cloudy. That's how we know there's growth in nutrient broth, in liquid nutrient broth, is because it becomes cloudy. So that shows bacterial growth. Now, in the second part, he boiled and sterilized the broth again. Inside of the building, he removed the flask briefly. So outside, there was 89% that showed growth. And when he did the same thing inside, 32% showed growth of bacteria. So what the first experiment told us is bacteria can come into a flask, even if it's open very briefly, just through the air, okay? So let's look back at the hypotheses that Louis Pasteur had. He developed this hypothesis that bacteria arise spontaneously in a broth. That was hypothesis A. Hypothesis B, which is the one I believe that he was going with, he believed that hypothesis B was going to be supported. Hypothesis B says bacteria in the air contaminate a broth. So our conclusion after the first experiment is that hypothesis B is supported because he just briefly took the stopper off of both flasks outside and inside a building, and he got bacterial growth after that. Okay, then he did another experiment, and here he used a curved neck on his flask. So again, instead of a stopper, he used a curved neck. So he boiled his broth to sterilize whatever was in there. So any bacteria that are in here are killed. And then he left the flask open to the air. And so we know air enters into this part of the flask, but what ended up happening is this prevented the growth of bacteria because the bacteria would collect at the bottom of this first dip in the neck, and they couldn't go up. They couldn't travel up the incline. So the air, that the bacteria that's collected here at that first dip in the neck, well, we were able to collect bacteria. I'm sorry. There was bacteria collected from right there, but no bacteria was collected from the dip at the top end that was closer to the opening of the flask. So that was telling us that the bacteria could not travel up. So no bacterial growth, and what he discovered was pasteurization. This is the concept behind pasteurization of milk and other liquids, is that you boil it, and then you boil it in a flask that has a curved neck that doesn't allow more bacteria to get in. Okay, let's go. Oh, I have to, sorry. Now, for prokaryote structure, whether they are bacteria or archaea, they're going to have a cell envelope, which consists of plasma membrane, a cell wall, and a glycocalyx, which can either be a kind of a liquid or slimy substance, or it can even be a solid, like a capsule type structure. But their prokaryotes typically have something outside the cell wall for extra protection. It's called the glycocalyx, and it also, the glyco means glucose. It causes the prokaryotes to be sticky, so they can stick to one another and to the surface that they're growing on. The cytoplasm of the prokaryotic cell contains the nucleoid region, which doesn't have a membrane. It's just an area where the DNA is found. Prokaryotic cells also have ribosomes, and cyanobacteria contain thylakoids so that they can carry out the light reactions of photosynthesis on their thylakoids. So that's only cyanobacteria. And then prokaryotic cells can have appendages, sometimes flagella for swimming, a conjugation pilus for exchange of DNA, and fimbriae are structures which help them attach to their surfaces and to each other. So here is a diagram. You can see the nucleoid region with the DNA in the center. The dots here are the ribosomes, which make proteins. And then if you blow up the envelope, just this section right here, I guess, the inner portion of the envelope is the plasma membrane. And then outside of that, you have the cell wall, and outside of that, you have the capsule. This particular prokaryotic cell looks like 3 flagella. They don't have to have any flagella, but if they do have flagella, it means they can move. This one also has a conjugation pilus, which allows for the transmission of DNA to a host, to another prokaryotic cell. And then fimbriae are these bristles that are attached to the surface of the prokaryote and to the capsule part. And the bristles help the prokaryotic cell attach to other cells and also to a surface. Reproduction occurs by binary fission. Prokaryotes reproduce by binary fission, which means to split in half. It is basically they clone themselves. They can sometimes reproduce as quickly as 12 minutes. So the generation time can be as short as 12 minutes, but it can also be longer. They have a high rate of mutations in their DNA. So they are haploid because they only have a single chromosome. So prokaryotes are haploid. They have a high rate of mutation. They can reproduce as fast as 12 minutes. This is a picture of a prokaryotic cell which is has almost completed binary fission and become 2 cells. The ways that prokaryotic cells The way they get variety in their genes, we call it genetic recombination. They don't technically reproduce sexually. There's not like a male and a female that, the male gives half and the female gives half of their DNA. But they can undergo a process called conjugation, and that is when one bacterial cell injects its DNA into another one through the conjugation pilus. So that means there's a donor cell and a recipient cell, and these are both prokaryotic cells. Can be bacterial or it can be archaea. But the donor cell passes DNA to the recipient cell. So this is from one bacterium or archaean to another. Then we can have transformation, and that is when a bacterial cell or an archaea, which is living, can pick up pieces of DNA from its environment that is left by prokaryotes which are no longer alive. So in other words, transformation occurs when a living cell picks up free pieces of DNA from other prokaryotes which are dead. And that DNA becomes incorporated into their genome, into their single chromosome. And then we can have transduction, which is when a virus passes bacterial DNA. It's like used as a vector. I can't get it to, there we go. It's when a virus passes bacterial DNA from one bacterium to another. So these viruses, again, are called bacteriophages because they only infect bacteria. Bacteriophages can carry portions of bacterial DNA from one bacterium to another. And in that way, the bacteriophage virus serves as a vector. So that's called transduction. Now, we're going to move on to the section 20.4 on bacteria. Bacteria are definitely more common. Of the two types of prokaryotes, they're the most common. Bacterial cell walls contain peptidoglycan. That's very important. Bacterial cell walls contain peptidoglycan. We can do a procedure called a Gram stain procedure, which we are actually doing in class. And that is when we use two different colors of stains. We use a purple stain and a red stain, which actually ends up looking pink. So we say purple and pink. If the bacteria are gram positive, that means their cell wall has a thick layer of peptidoglycan, and we say they're gram positive. They are going to appear purple under the microscope. They will absorb the purple stain. There we go. I don't know why it's not. If they're gram negative, then they will not absorb the purple stain. They have a thin layer of peptidoglycan and it won't absorb the purple stain. So they're going to actually appear pink. It says red, but when you see them under the microscope, you'll see they actually look pink. So if they're gram negative, they're going to stain purple. If they're gram positive, they're going to stain purple. And if they're gram negative, they're going to stain pink. And you can see the gram stain procedure is shown here. The first thing that we do with our microscope slide where we have a sample of bacteria that we have that has undergone a heat fixing procedure. So after we heat fix the bacteria to the slide, now we're going to apply crystal violet, let it sit, and then we're going to wash it off with water. And then we're going to apply iodine, and that helps the crystal violet absorb into the cell wall of the gram-positive bacteria. So gram-positive bacteria need the iodine to help fix the crystal violet stain to their cell wall. So if they're gram-positive, they'll absorb that crystal violet. Then we use an alcohol wash called decolorizer. It's just ethanol, and that's going to wash all of the purple stain off of the gram-negative bacteria. So we'll have to stain them with safranine, which is a red dye, and they're going to end up appearing pink under the microscope. You can see here, you can see that some look, there's some purple, which actually look like endospores. So most of these are pink. So the bacteria in this sample that you see in picture B are actually gram-negative. Now, bacteria, now that we have stained them, we can see their different shapes, and that can help us to classify them. If they're spiral-shaped, we call them spirilla. If they're rod-shaped, we call them bacilli, and if they're round, we call them cocci. So here you can see a spirillum. You can see a bacillus. This is E. coli, Escherichia coli, which is a typical rod-shaped or bacillus-shaped bacterium. And then here you can see a chain of round bacteria called cocci. Each one is called a coccus. This is Streptococcus thermophilus, and because it's in a chain, that's where the strepto comes from, but the shape is coccus or round. And we also have 4 different types of metabolism in bacteria. And before we get to the 4 different types of feeding, we are going to talk about the whether or not they require oxygen. So if they do require oxygen and they must have oxygen, they can't grow in the absence of oxygen, they are obligate aerobes. So they're aerobes if they require oxygen and obligate if they can't grow without it. We are obligate aerobes. Humans are obligate aerobes. We must have oxygen to survive. So if a bacterium is an obligate aerobe, it must have oxygen to survive. If it's an obligate anaerobe, it cannot survive in the presence of oxygen. So it must have the absence of oxygen to survive. The bacterium that causes botulism, gas gangrene, and tetanus, botulism is a disease you get from eating improperly canned foods. So it's like a stomach infection, a stomach disease. So obligate anaerobes will die in the presence of oxygen. But then we have facultative anaerobes, and they prefer to be without oxygen, but they will not die in the presence of oxygen. So they can survive in the presence or absence of oxygen. Okay. Now, the types of feeding you can have, just like a plant, you can have bacteria which are photoautotrophs. They use photosynthesis in order to make their own food. So photoautotrophs use sunlight for their energy, and they convert carbon dioxide in the air into organic compounds like sugar. Now, if they're oxygenic, they're like plants. That means they also produce oxygen through photosynthesis. But if they're anoxygenic, like the green sulfur bacteria and some of the purple bacteria, then they do not. They are found living in oxygen-poor conditions, and they don't provide oxygen. Okay, so we have cyanobacteria, which isn't listed here, but the cyanobacteria are the bacteria which carry out photosynthesis like plants. They're oxygenic and photosynthetic. Then we have chemoautotrophs, which is unlike plants and it's unlike animals. Chemoautotrophs use inorganic compounds, some type of inorganic compound to obtain energy rather than light. So they break down or oxidize inorganic compounds for energy. And then that energy is used to convert carbon dioxide into food. We have some that live in deep sea vents, like 2.5 kilometers below sea level, which is at least a mile below sea level. So that's where it's very hot, very acidic, and they break down sulfur compounds a lot of times for energy to convert carbon dioxide into food. Okay, so if it's an anoxygenic and if it's anoxygenic and photosynthetic, it doesn't make oxygen. If it's oxygenic and photosynthetic, it does produce oxygen. So the oxygenic photosynthetic bacteria are cyanobacteria. The ones that are anoxygenic and carry out photosynthesis are sulfur bacteria. They're green and purple sulfur bacteria. Then we can have heterotrophs. If they are chemoheterotrophs, they are like humans. That means that they break down the food that they eat and use that to, well, that is their source of food. In other words, they consume food. They don't make food from carbon dioxide. But there are also chemoautotrophs, and I don't see that listed anywhere. There's chemoautotrophs that break down chemicals for that's chemoautotroph, I'm sorry. There's chemoheterotrophs, and what I don't see, I apologize, what I don't see is photoheterotrophs. There are heterotrophs that take in or consume food, but they get their energy to break down that food from light rather than from breaking down chemicals. So chemoheterotrophs consume food and they get their energy and their nutrients from consuming food. Okay, so I'm going to stop here. We'll start back on this slide because we didn't get to cover the whole thing, but I'm going to stop here